Very good morning, everyone. Today, my name is Ullas KANNANTHA. I'm a second-year master student in neuroscience at Bordeaux Neuro Campus. Today, I'm going to present our work, Disruptive Spontaneous Network Activity in the embryonic SOD1 G9-3 mouse model of amyotrophic lateral sclerosis. This work was done under the supervision of Anthony Sarnacki and Pascal Branchot, professors in neuroscience. As you all might know, amyotrophic lateral sclerosis is a devastating degenerative disease affecting cells in the spinal cord as well as the brain. Now, looking at genetic epidemiology of ALS, we find familial ALS to be around 10% of total proportion of ALS, out of which the SOD1 mutation accounts for the second most prevalent case in the European population, and it is the most prevalent case in the Asian population. These data highlight the importance of looking at SOD1 mutation in context of ALS. Now, ALS is perceived to be neurodegenerative disease. However, there is growing body of evidence showing embryonic alteration in ALS mouse model. For example, Martin and others showed hyperexcitability at E17.5 in SOD1 G9-3 mouse model, and another team showed altered chloride homeostasis at embryonic day E17, also in the same model. These data highlight it's important to look at development in context of ALS. Now, looking at development itself, we observe spontaneous network activity. First of all, it is ubiquitously observed. It is observed in the spinal cord, cochlea, retina, hippocampus, and cerebellum. And it is synchronous firing of ensemble of neurons. And another important point is network driven. I'm going to play the video here where you can see the spontaneous network activity in turtle retina spreading across the whole retina. And this is one of the important features of spontaneous network activity. Now, looking at spontaneous network activity in spinal cord, we find it during embryonic stage. And it is characterized by giant depolarizing potential, and this potential can last for about 2 to 10 seconds. And it plays a very important role in neurogenesis, synaptogenesis, and axonal pathfinding, to name few. Now, the Landmesser team in the United States showed that disrupting the spontaneous network activity, what they did was they increased the duration between two subsequent GDPs, and they showed that disrupting the spontaneous network activity leads to disrupted motor neuron pathfinding. And the same team also showed that disrupting the spontaneous network activity also leads to disrupted motor neuron migration. This shows spontaneous network activity is important for normal development of motor neuron. Now, looking at crucial embryonic time window in mouse, we find spontaneous network activity between E11 and E14.5. And as you can see, during this period, important phenomena like synaptogenesis, migration, and neurogenesis is taking place. And for the sake of the study, we look at last 2 days of synchronous firing of spontaneous network activity, that is E13 and E14.5. So considering all the things that I said so far, we came up with the hypothesis that if SNA is altered in the early embryonic development of SOD1 G9-3 mouse model of ALS, it may contribute to the subsequent motor neuron dysfunction that we observe in this mouse model. To answer this question, we performed whole cell patch clamp recording on lumbar motor neurons taken from E13 and E14 embryos, and we looked at giant depolarizing potential GDP in current clamp and giant inward current jig, which is its voltage clamp counterpart. So the first task, we look at embryonic stage E13.5. So we measure period. Period is the duration between two subsequent GDPs. In the literature, you might also see it as inter-episode interval. We did not see any difference between SOD and wild type. Next, we looked at surface. Surface, the area under the GDP, it was also no difference between SOD and wild type. And we looked at the amplitude excluding the spikes, it was also no difference. And the duration of single GDP was also no different. At bottom line, at E13 in this mouse model, we did not observe any alteration in spontaneous network activity. We move forward and we look at E14.5. Firstly, we observed different types of activity. It's not a surprise, people have already characterized that. We observed short duration GDPs. We observed long and short duration GDP in a single recording. We also observed in a recording where we had erratic GDPs, and we also had recordings where there were no SNA. So this is the distribution we have in wild type. So what's surprising here is this distribution of different types of activity was significantly different in SOD and wild type. To highlight one of them, we observed more short GDPs in SOD compared to wild type. And the second surprising result is we looked at capacitance. Capacitance is an indirect way to measure the cell size. We observed significantly smaller capacitance in SOD compared to wild type, meaning the SOD motor neurons were significantly smaller compared to wild type at embryonic stage E14.5. Next, we looked at spontaneous network activity properties at E14. Firstly, we looked at period. We observed higher period, longer period in SOD, and the distribution was pushed towards the right, as you can see here, and we observed surface to be smaller in SOD, and we also observed the amplitude to be smaller in SOD. However, the duration was no difference between SOD and wild type. So the longer period between two subsequent GDPs means there is less SNA at E14.5 SOD1 G93 mouse model. So just to summarize what I've talked so far, so the size of motor neuron that we observed at embryonic stage E14.5 was similar to that of E13 wild type. And the types of motor neuron that we observed at E14 was also similar to that of E13 wild type. This shows there is delay in development of at least 1 day in this mouse model. Next, we wanted to know a molecular mechanism that could answer the observations that we had so far. We looked at persistent sodium current, persistent sodium INAP. Persistent sodium current has been shown to be important for contributing to the patterning of spontaneous network activity. And the previous study showed that by blocking the persistent sodium current, they observed decrease in giant inward current amplitude and also increase in period. This is the similar kind of observation that I had at E14 SOD. This led to our hypothesis that persistent sodium current is decreased in E14.5 SOD mouse model. So to answer this question, we first pharmacologically separated out the persistent sodium current, and we measured the current density, that is the persistent sodium current normalized to the size of the cell. We also looked at peak potential, the potential at which we observed this peak inward current. Surprisingly, there was no difference between SOD and wild type. So bottom line, the persistent sodium current is not responsible for the observations that we had. Next, if you remember in the introduction that I said one of the important role of SNA is synaptogenesis, and knowing that we observed delay in development at E14, and also knowing the fact that SNA is synaptically driven, we came up with the hypothesis that synaptogenesis could be downregulated at E14.5 SOD. We looked at synaptotagmin 2, which is a crucial marker for synaptic activity, and we indeed found a significant downregulation of SOD in synaptotagmin 2 in SOD compared to wild type, and this downregulation was about 20%. So to summarize the whole talk, firstly, we did not observe any alteration in spontaneous network activity at E13.5. However, we observed clear alteration of spontaneous network activity at E14, and motor neurons were significantly smaller, and it was similar to that of 1 day previous, like E13. And we also observed downregulation of synaptotagmin 2. This data shows there is delay in development and also decreased synaptogenesis. And as a future direction, we would like to study the whole crucial embryonic time window that I talked about. Now, importantly, all the data previously which had showed embryonic alteration in ALS mouse model, along with this study, point us in the direction of neurodevelopmental aspect for this ALS. Of course, we have to do more studies and across different mouse model of ALS to make a concrete statement. Now, I'd like to thank people who are directly involved in this project, William, our engineer in the lab, Dr. Anthony Sanaki, Dr. Pascal Brancho, who is with us today. And also, I would like to thank the funding agency, ARSA is one of them. Thank you for that. And I thank all of you for your attention and happy to answer a few questions.
Thanks. Thank you. Other questions? Carolina.
Thank you very much. I had a question regarding the hypothesis that may be going on here. Do you think that motor neurons in the SOD animals are born later than in the wild-type little mice, or would it be that they are born at the same time, but they mature more slowly?
If I could take a guess, they're born at the same time, and as we know that spontaneous network activity is crucial for normal development, because of this alteration, there is delay in, or the difference in size that we see one of them. Mm-hmm. So, but we didn't look at motor neuron, whether they were born at the same time or not.
It could be trying BRDU stainings at key moments to see when those cells are born.
Definitely.
Thank you.
Actually, I have one question. Did you check whether the developmental death of motor neuron which happens also around E13 and E15, if I remember well, is modified?
We did not look at the death yet. Maybe we'll have to look at it in the future.
Okay. Other questions? If not, in the interest... Oh, well...
This is a very naive question. Is SNA an equivalent of fasciculation? Or?
I really do not know. maybe my supervisor might have some thoughts on it. Maybe we can sit and discuss it after this talks.